Enzymes, cool. When we were talking about proteins, which we've been doing for about a week now, we were saying earlier that they sort of occupy two roles within the body. A lot of them are structural, they give you three-dimensionality. The structures that like hold your cells together, because you're multicellular and your cells stick to each other. Your cells are sort of stitched together with protein strands. The cellular scaffolding that you have is all protein-based. Your cytoskeleton is a protein. You have proteins all over the place with them. So if we suck all the water out of you, the mass that we have left, about half of it will be protein. The same goes for broccoli. So people say, like, how do we get a lot of protein in my diet? It's like dark leafy green is the same as always. Everything has scads of protein in it. Everything alive has scads of protein in it. The other thing that strands of amino acids do when they actually catalyze a reaction, we refer to them as an enzyme. So they're not building structural support. They are sort of the worker bees of the body making reactions happen at a known and given rate. And the rate, if we wanted to come up with a term and what is the unit of what rate enzymes work at, they work at the rate of your life. You can only live your life as quickly as the rate of your enzymes can do things. So it's like how fast do you perceive time as moving, right? That's a function of enzyme activity and how fast enzymes work, which is kind of wild, right? I mean, you can feel time moving, right? Right? Can you feel it going forward? So now it's now, but now it's now. But now is later than then, which was the now then. But that's not the now, because now is the now, but now it's a different now. Right? That's weird, isn't it?
Right.
Because it's always now, but it's always also then. So when the hell actually are you?
Now.
But now you're.
Then.
No. But the now that's now refers to the now that you were then. But now you're now, so you're not that person any longer, which means you get a free pass on everything that you do because you're not the same person anymore. And clarifying that is a problem for the later us.
Tomorrow never comes.
It never does. It's always now. So just be happy, right? So this whole, I need to do the right thing. I need to get the right degree. I need to get the right job in order to get all these things. It's all a bunch of bullshit, right? You have no confidence where anything is going to happen. You're not actually in control of any aspect of your life whatsoever and what happens to you. What a gift. Just do what brings joy and don't be an asshole to people. Treat waitstaff kindly.
Yes.
Be nice to servers.
Very.
Please, tip well and be kind. They're just doing their damn job, for goodness sakes. So this rate, so it's like, how do you actually feel time moving forward? Well, you have these fun little nervous structures in your body kind of scattered around a little bit. They're nervous tissue, and they kind of spontaneously generate a nervous signal, almost in a clock-like way. Every second or two, they send a little tick. And that happens at the rate that enzymes are functioning. And that constant tick of a couple of neurons in your body gives you this neurological sensation of time. It's not a real thing, but it's like you sort of build this. There's this thing called time in your perception of how the universe works. Everything already happened. We're just waiting for our neurons to catch up with the inevitability of the universe, which is already played out. But I mean, when you took philosophy in 5th grade, you talked about this stuff, right? So it's fine. Good.
Yeah.
Yay! So we're catalyzing. Reactions. What does that mean to catalyze a reaction? It does not make magic. They don't make magic happen. What I mean by that is that the reactions that enzymes do are reactions that can happen on their own but what the enzymes do is increases the rate or likelihood of the reaction happening. So, it's not, they can't do things that were not a possibility. So what enzymes do is they adjust the odds in favor of the reaction happening. So what this means, extreme cases, just the construction of a glucose molecule from a lot of carbon dioxide. Photosynthesis does this very well, right? That's a sequence of several hundred interacting, interoperable enzymes that are making that reaction happen. But, conceivably, there's a tiny fraction of the number of stars, 1 over the number of stars in the universe kind of chance that you could get a bunch of water molecule and 6 molecules of H2O, and they could hit each other, or CO2, and they could hit each other in just a particular way that a glucose molecule will form. It is extremely unlikely that glucose is going to form in the environment without photosynthesis. Are you with me on that? But these molecules bounce around all the time, or the parts of here, it could conceivably happen. What the enzyme says is this is never going to happen at a rate that's life-giving in any sort of way. So we'll kind of come up with a biochemical pathway structure where each part of it does a little, this one grabs a molecule of CO2, this one grabs another molecule of CO2, this one takes it from that one, attaches it to that one, this one adds a water molecule in and strips a hydrogen out so we can bounce it. Then it goes back and grabs another CO2 and attaches it onto that, right? So you get all these enzymes that are working together, each doing a tiny part of a sequence that together take CO2 and water and build an actual 6-carbon glucose molecule out of it. So that could happen spontaneously on its own at an extremely improbable random rate, which is probably not going to happen in our lifetimes. But so it's not making magic happen. There has to be some probability that could happen on its own for the enzyme to work. But the enzyme sequence can significantly increase the odds and therefore rate of it happening. Reaction. And the way that it does this is a model that we refer to as induced fit. Induced fit. Sometimes if you put together IKEA furniture or do any sort of construction whatsoever, simple construction, what you find is that if you don't have the proper tools to do something, stuff can take a very long time. Or you can go to Home Depot, buy the right tool for $3.99, and the thing's done in 5 minutes. Did you ever witness that or have that happen? It's like the right tool makes things happen quicker. It's almost the exact same phenomena. So if we think about an enzyme, here's a big 3-dimensional structure, there's coils in here, there's folds, there's more coils working together on this thing. Cool. Here's a big fat enzyme. We made it out of the long, we had a DNA strand, we separated it, we made some messenger RNA, we sent it over to a ribosome, we translated this into primary protein sequence, and it folded into a quaternary or tertiary structure that now is 3-dimensional shape, right? So this is like all long series of amino acids that are folded upon themselves in a way that gives us something that looks like this. So here's our enzyme. What this enzyme seems to do, it takes molecules that look like that and molecules that look like that, and it turns them into a molecule that looks like that. It turns out this little block fits into that space right here. This little thing can physically fit into this space right here. When they fit in there, this enzyme will actually deform and the two sides will spline together, splang, and what poops out is one of those. So this region right here, where all that action happens, we refer to as the active site. Does that make sense? Perfect. My skin is really dry by the end of the day. It's chalk. It sucks the moisture. Need to moisturize.
Do you want moisturizer?
No, I'm good for now, thank you. We might talk later, though. Might talk later. All right. Any questions about this so far?
So the whole squiggle thing is identified?
Yeah, this whole mess down here is an enzyme. What if this is a reaction that's happening in our body and we want this reaction to stop? Let's say this is a reaction that is part of a cascade that triggers an immune response and we're going into anaphylactic shock because we ate some peanuts and we're allergic or something like that. We need to make this reaction stop right now. So we can take something, injectable, breathable, inhaler, or a pill or something like that. And what it is that we take can actually, like an aspirin or prednisone or something that we call pharmaceutical, we take this little thing, and this little molecule is shaped like that. You bought this from Pfizer or GlaxoSmithKline or something like that. This thing, you take it, it lodges right there in the active site of that enzyme. Now, this cannot fit into that location. We have engaged in what we call competitive inhibition. This thing we took is competing with this active site, with the actual substrate of the reaction. This would be called a competitive inhibitor. Many medications are competitive inhibitors. I think either acetaminophen or ibuprofen, one or the other, is a competitive inhibitor. It literally blocks the active site. There are some toxins that do this as well, like spider venom and things like that. They can actually block part of the active site in places that are really important for you to not die. So that's one way we can artificially control a reaction. There are others. Another way is if we have, can I erase some of this? Anybody else? All in favor?
Yes.
Any opposed?
No.
I think you guys have it.
So the square and the triangle represent specifically?
They are referred to as substrates. They are the raw materials that are getting acted upon by the enzyme. So the enzyme is interacting with the substrate or substrates to combine, attach, take apart, whatever it is that it's doing. We can also have non-competitive inhibition, where we have, here's our enzyme, here's our active sites. You with me so far? And you take this thing that looks like this. And when you consume or injected by or inhale this thing, it won't attach where it is. It'll slide in right here in between. It's not directly competing with the actual active site of the enzyme. However, by hanging out and checking in right there, if the two components actually do land here, it prevents the active site from deforming to bring them to come together, to join. So like it's literally getting in the way of the active site actually functioning, not by directly competing with the location where the substrates attach, but by preventing some other aspect of it. And it can even like attach down here somewhere in a weird way, which does literally change what that active site looks like by changing the folding of the protein itself. Either way, this reaction is going to stop. It's not going to progress into this thing because we've mucked with the active sites, either directly or indirectly. So what do you think about that? So many, if not most, pharmaceuticals that we take do this in one way or another, either competitive or non-competitive inhibition, to either increase the rate of a reaction happening or stop a reaction from happening in one way or another. Anti-itch cream or something like that, that will do this one way or another. Questions, thoughts, comments? Go ahead.
Can you repeat the non-competitive?
Yeah, so there's still a molecule that's out there that's being introduced to the body in one way or another, and it's interacting with the protein, but not in the active site directly, but in a place that does affect the function of the active site, either by preventing the molecular motion within the protein so the thing happens, or by just like attaching down here and like literally changing the shape of the active site by changing the protein fold itself. Which is neat. Go ahead.
So the small triangle that you made and that separator thing, what are those specifically called?
These things?
No, like when you made like this.
This thing?
Yeah, that or the other one that just didn't let the.
We would call them we would just call them, the general term, we would just say they're an inhibitor, an enzyme inhibitor.
Okay.
Yeah. And this assumes that the enzyme is out there and it's functioning all the time if the substrates are present. The opposite can also be true. Another thing we can do is, it's easy to draw enzymes. You just kind of, it was the first thing you ever drew in your life was an enzyme. 6 months old and you had a crayon, you drew an enzyme. Right. Let's say we have our same, I did the other way, didn't I? This thing. And it makes a molecule that looks like that. Good, good. So this is our reaction that we want to have happen. We want to take these two substrates and combine them into this product. We would also call these reactants. You could call them reactants in a biochemical pathway. And here we have one active site. And here we have another. This fits into here. This does not fit into here. It's the wrong shape. Okay? So maybe what you want to do is have the enzyme out there and present all the time in your body, but not functioning. But your body can release something that you would call an activator. And an activator, little star-shaped thing, has this fun thing. It can swoop in, attach onto the enzyme somewhere, and when it does, contorts the shape so that it flips in to look like this. Now it works, and you get the product. So in the inhibition the enzyme works and something prevents it from, and then something sort of breaks it or stops it. With the activation, the enzyme doesn't work, but then your body releases something that turns it on to make it work, as long as that activator is present. What do you think about that? So the enzymatic reaction that we're working with in lab this semester is called catechol ACE. Catechol ACE. You know it's an enzyme because it ends with ACE. The reactants are molecule called catechol, which does this curious thing, and I'll tell you what it is. And it takes oxygen and it turns into some melanin and melanin precursors. Melanin, which is sort of a medium darkish orangey color. It looks like, and other things too, right? It's not quite as simple as it makes black people black, right? But brown people brown. That's part of it. Melanin is part of it. Pure melanin alone looks like spray tan. It looks kind of like spray tan, right? You see it work. This reaction when you take an apple and you cut it in half and you let it oxidize and it gets kind of dark, kind of this darkish, or it's not totally brown. It's like darkish orangey color. You with me? That is this reaction. And it's also the reaction that you see in spray tan. I'm looking at you, former president. Yeah, so like if you get like a spray tan or an artificial tan, it's like they're literally catechol in you, right? So it's like, it's close, it's not quite the real thing. There are other things in the skin that gives us the darkness and variation that we see in skin tone. But this is part of it. Good thing. Well, this reaction happens at a rate, go ahead.
I was gonna ask, when we sit in the sun, is our skin oxidized?
When you sit in the sun and you get darker, more of this reaction is happening, and it darkens us, right? And that does require oxygen to be present, but it also requires something else to be present. It requires an activator. That activator is copper. Right? This reaction only happens in the presence of copper at the rate that's appreciable. So sometimes if you take a multivitamin, one of the weird components of it is like copper. What do I need copper for? That's what you need copper for. You know, it's
So you're saying if I want to be really tan, eat copper?
If you want your melanin to work, you need copper. What you probably get just from eating dirt. Eat some vegetables from a farm. You can find them in plants, right? I mean, animals eat copper, and then we eat the animals. You don't need a lot of it. It's like, oh my God, my professor said I need copper, so I'm going to find some old pennies and suck on them all day. Like, that doesn't quite work that way. But I mean, you need copper for that reaction to go.
And so,
Good news, you have it though.
okay, weird question. So people
Unlike any other question.
people who have I guess are a little darker, who are more easily to be able to tan rather than people who have more like whiter complexion, who burn usually. Is that because of the lack of copper? Usually.
Usually, no. It's usually the lack of the
so what's the difference between
catechol
people
production.
who don't burn? Like for example, I never burn. Like I just.
Yeah, it takes a lot of sun for me to cook. However, my sister, who looks nothing like me, who I swear, if she didn't look just like my dad, would look like, I'd say, you look like the mailman, more than you look like me. Mom, is anything like to say? Dad, is there anything like to say? My sister goes from white skin to red, back directly to white. Do not pass tan, do not like $200 or anything. It can get pretty dark in the summer.
Mhm.
It can get pretty dark, right?
So what would be the difference? It's because one has more.
It's the substrate.
Okay.
Yeah, it's the production of the substrate. And so what we see in the variation in skin color throughout the human population we see different levels of production of the precursors of melanin.
So that's what basically protects you from your burns, right?
Well, anybody can get a sunburn. Anybody can do that, right? The skin tone that we see around is usually less about protection from burning and more about vitamin D production and the ability to absorb sunlight to get vitamin D. If you live in the northern hemispheres, you need a lot. So we need to make sure you're nice and white. Kind of like in Scandinavian countries, things like this, you know. Yeah. So there's this whole huge history of like following the natural history of skin tone, right? And how it originates and where it goes and what's it for and all this kind of stuff. It's pretty fascinating. But vitamin D seems to be a big part of it, production of vitamin D. It's like if you're white in this classroom, you probably have a vitamin D deficiency.
Thank you.
Go outside and get a little sun, you know? It's sort of how it works. So this is the reaction, right? So when this reaction is not happening, you're just going to get like this colorless or very, very faint orange solution. The more the reaction progresses, the more darker orangey brownish the test tube will get, which is kind of neat. And so we'll fool this in the lab a little bit. Probably today. So there's competitive inhibition and non-competitive inhibition. When we think about life on Earth, a lot of times, though, we think about regulation, right? We want things to be homeostatic. Have you heard the word homeostasis before? If your body temperature starts to go up, your body will then attempt to bring it back down again. If your body temperature starts to drop, your body will start doing things to bring it back up, right? You maintain these static levels of biochemistry with regards to physics throughout the course of your day, right? You regulate stuff. When your blood sugar starts to go up, you produce insulin to bring it back down. When your blood sugar starts to drop, you produce glucagon, which brings it back up. I mean, that's our liver example that we've been talking about earlier with sugars. All this kind of stuff happens. When you start to get a lot of triglycerides on your body, those lipid cells produce leptin, which is an enzyme / hormone that suppresses hunger response. When your body starts losing a lot of triglycerides, you're not producing that leptin, and you feel hunger more strongly, and you have more of an appetite. So it's all throughout your body, all of these reactions are trying to maintain these basic static levels of functioning in one way or the other. You know, does that make sense? So the question is like, how does your body maintain that stasis, that homeostasis that we see? We can almost think about it as the HVAC system here on this campus, right? When the temperature drops below 60, the heaters come on and the temperature goes back up. When it gets above 75, the coolers come on and the temperature goes back down. So we like the temperature in this building. We just want to keep it a nice, level, constant, steady rate. That makes sense, right? You hover around 98.6 degrees. Blood sugar is about 80 or 90 deciliters per, milligrams per deciliter, right? So, I mean, you have all these different, your blood oxygen is usually about this, your blood CO2s, your pH is usually just about this, your blood urea count is usually just about that. All these parts of your body that you can measure that are homeostatic and regulated one way or another. How does that work? It works like this. I'm going to continue, I'm going to draw a different protein. Let me erase this too. What would happen if you had a series of enzymes that stretch out sort of in a process? So you have enzyme A, which produces something that is a reactant for enzyme B, that produces something that feeds into enzyme C, that produces something that goes into enzyme D, and you get a product. And over here you have reactants. We can draw our square or our triangle again. And over here, we'll have our product, which is our square attached to our triangle. Does that make sense? Series of 4 enzymes daisy chained together. This one does the first part of the reaction and passes it. This one does the next part. This one does the next part. This one does the next part. And when all 4 of these are working, you go from these 2 separated things to these 2 molecules attached to each other. This is the product of that reaction. Does that make sense? So far, so good? Perfect. What if enzyme A had a site on it right down here in the bottom of it? And the actual active site where the first part of the reaction was up here. And this is a site down here where the inhibitor site where an inhibitor can attach. And the inhibitor of the reaction is the product of the whole thing. If you don't have any of this in your body, this step won't be inhibited, and you will start making this stuff. You will start making this stuff until it builds up in the body. After you have some of this in the body, it will start inhibiting this part of it, and you will not continue making some of this. When this drops in the body, you start making more of it. When you start making enough of it, production starts to tail off. What do you think about that? So it's like the actual product acts as an inhibitor earlier in the reaction series. This is called feedback inhibition. What molecule acts as a feedback inhibitor that shuts down the release of glucagon? It would be glucose. You can have a molecule like glucose that when it attaches to the receptor, increases the release of insulin, right? Through the actual reaction product acting as an inhibitor on the enzyme series, you can maintain homeostasis, which is wild. The presence of the product shuts down the production of the product. So if that drops or becomes non-existent, you start making more of it because it's no longer inhibiting. Once you get to a level where it starts inhibiting over here, you stop the production of it, which is neat. So let's say you had an assembly line where you're going to make a bunch of pancakes, all right? And there was a scale that the pancakes dumped out onto. And when you get 1 pound of pancakes on the scale, it flips a lever that shuts off pancake production. So if there's no pancakes being made, the scale's not detecting mass, you're going to make pancakes, right? When you hit 1 or 1.1 pound of pancakes, the switch flips and then pancake production starts, correct? And then Brooke says, I like pancakes. Brooke comes up here with a fork and some maple syrup because it's fall and eats some pancakes. The mass goes down to half a pound of pancakes. He had a lot of pancakes. That's a few really big pancakes. So now we have half a pound of pancakes because Brooke ate some. What happens to our production line? Yeah, it's under a pound. It's going to turn back on. It's going to produce more pancakes until it gets back up to a pound. So here is like we have this production series where the actual end product regulates whether it operates or not through feedback inhibition. Does that sound good? Does that make sense? So enzymes work a lot of different ways. If we already know how proteins are made through transcription and translation. We started our class this way, correct? We have a series of DNA, we have to split it, we have to make a messenger RNA sequence out of it. Did I tell you about did I tell you my corn joke? It's the corniest joke I have, right? About I went to the farmer's market and I got went up there and I said, can I get some of the pirate corn? And they said, pirate corn? I said, yeah. The punchline is,
No.
it's a buccaneer. You still not think that's funny?
Yeah, it's a bug.
You guys were like totally checked out on me lecturing, so I thought I would tell you some jokes. You doing okay?
Yeah.
I'm seeing blank faces. Can you do some arm circles? Go ahead. Yeah, totally. Like you did one. Yeah, I think you're suffering from low blood sugar today. But yeah, good call, Ron. So go forward, so go back.
Sorry.
Who has a better joke than the Buccaneer joke? Anyone?
Ooh, I got one.
Ooh, go ahead.
So, an American and a Russian are having a political argument with each other, right? And the American says, things in Russia are so strict. If there's something that I don't like, I can go up to DC, I can walk up to the White House, I can slam on the president's desk and say, Mr. President, I don't like how you're running the United States. And the Russian says, I can do that too. American says, really? He says, yes, I can walk up to St. Petersburg, I can go up to Vladimir Putin's desk, I can slam on it and say, Vladimir Putin, I do not like how the president's running the United States.
That's funny. So laughing more at yours than mine, so we can't tell any more jokes. Sadly. A lot of ways to control this, but the last thing before you take a little bit of a break and then do some lab stuff. Going from a DNA strand to a functioning protein is not something that happens in the fraction of a second. Correct? I mean, it's a rate. There's a lot of things that have to happen. You need to make a strand of messenger RNA. You have to send it to a ribosome. You have to make translation happen. You got to get your codon table out. And you have to do all this stuff, right? What if it's like, oh my God, something happens that I need to act very, very quickly. I need to hurry up and make enough of that protein so I can do something about it. It's not going to be made quickly enough. You with me?
Yeah.
Well, how your body works most of the time is by making all of the proteins and enzymes that you need in advance. They're already in their body, they're already out there, they're all doing their thing, but they are either inhibited or they're waiting for an activator. What your body will oftentimes do is store the activator in cells nearby the active site of the enzymes that you're trying to activate. When the emergency happens, you don't suddenly start rushing into production of enzymes. All you have to do is release the activator that will attach to the enzyme that's already made and turn it on. That's how you can do things quickly. So you make all the enzymes in advance, and then all you have to do is activators or inhibitors to turn them on and off, which you can do very, very quickly because you're also making those as well, but you're just keeping them separated from each other. So your life is not about making all this stuff when you need it. It's about releasing the components to turn enzymes on and off as you need it, which happens way faster. So it's like, why are we talking about this dry ass lecture about activators, inhibitors, active sites, inhibitory sites, feedback inhibition, and homeostasis? It's like, well, that's how you do it. That's how you do it. If you had to wait through the entire production process for enzymes when you needed them, it would take a month before you could actually do the thing that you needed to do. So you make all the enzymes, you make all the activators, all you have to do is release them when it's time. That sound good? Make sense? Good, cool. Take like 20 minutes, stretch out, grab a snack. We're gonna learn some skills in the lab. What I noticed from 14 years of teaching is you can't learn skills and learn biology at the same time. So in the first part of lab, we're going to learn a skill. And then my hope is in the second part of lab, we're going to use that skill to actually do a thing. So we're going to learn the skill first. Sound good? See you in 20 minutes.
I think that means you could ask him, but I don't